Okay, good morning. Today we're going to talk about some examples of DNA testing. And as a preliminary, I would like to remind you it's going to rely on the same natural DNA molecules that we acquire from a person, as well as those that are made by machines. Oligonucleotides is a general term. And some of this is going to involve PCR, but other examples are going to require specific oligonucleotides that are intended to detect the actual mutation that we're looking for. So the oligos are used for two different purposes. One, to PCR a region of the DNA that we want to look at, and the other is to actually identify some specific mutation that might be present in that DNA. Okay, so remember that the bonds holding the nucleotides together in a phosphodiester bond are very strong and covalent, whereas the ones that hold the two strands together are non-covalent and easily broken by conditions like high temperature, etc. This is a useful trick because once we've separated them, we can then hybridize them to something else that we put in that's complementary to the strand that we're looking for. Okay, so here's our little picture of our probe hybridizing to its complement. Okay, the target DNA would be DNA which has been PCR amplified from a person, for example, and the fluorescently labeled probe is going to be something that's complementary to that DNA that we're going to use the probe fluorescence to look for and see the location of that molecule in some other situation later on. Okay, so most often, in general, the DNA that is being hybridized is not a natural DNA. It's going to be an oligonucleotide that's made by a machine that were made to the specifications of the person who wants it. So it's going to be something small in the range of 15 to 50 nucleotides. It's going to be single-stranded, and it's going to be made to be complementary to the sequence that we're trying to bind to. Okay, PCR primers, of course, are examples, but probes are also another example of the same thing. And it can be confusing because they're both synthetic oligos made by machine, but they're used for very different purposes lots of times. Okay, so the little dot is to let us know that we can see it later. They used to be radioactive, but now they're mostly fluorescent, as we'll see. Okay, so DNA testing for diseases can be straightforward and simple, but sometimes it's complicated by the fact that there are many different mutations that can cause the same disease. Genes are big, they've got plenty of places where mutations can occur, and some genetic diseases are really quite heterogeneous in terms of the actual mutation that you're looking for, and that can complicate the situation hugely. But we're going to start at the very beginning, historically, with the sickle cell allele of the hemoglobin beta chain versus other beta thalassemias. Okay, this is historically interesting, and the method I'm going to show you is out of date, except in some sense conceptually. We know what mutation we're looking for in the case of the sickle cell. It's always going to be one particular mutation, one particular allele, and so we know what exactly we have to what we have to design in order to be able to detect that mutation in somebody's DNA. And then we're going to sort of move forward in time and talk a little bit about example of cystic fibrosis, where there is actually one major mutation, but many, many other possibilities. And for that case, we're going to have to set up situations where we're looking for different mutations, many of them in the same test, hopefully. Okay, so here's the idea of our sickle cell allele and the what's called the allele-specific oligonucleotide. Now, for some reason, the wild-type allele is called beta A, beta referring to the beta chain of hemoglobin, and A referring to the wild-type allele. The beta S allele is the one that causes sickle cell disease, and it's a single nucleotide change relative to the wild-type, where we have a GAG codon, which now reads GTG, which gives you a missense mutation, which affects the function of that protein. So we're really looking for a needle in a haystack here. We're looking for an A versus T change at that one codon, at that one position in that one gene in our whole genome. Okay, importantly, this oligo is only 19 nucleotides long. And so if we try to match that to something it's not completely homologous to, that mismatch will destabilize the hybrid. So this is set up in such a way that only the exact match can actually bind to the target DNA the mismatch is sufficient to destabilize any binding that does occur. And in that way, we can look and see what allele is actually present based on that difference in the hybridization ability. Okay, so our traditional old dot plot test, we're going to obtain DNA samples from people of various genotypes, and then we're going to apply a PCR test simply to make enough of that region of interest that it's easy to detect any mutations that are present or whatever alleles are present. So we're going to PCR the regions, like set up primers, maybe 100 nucleotides on each side of the actual nucleotide we're trying to see, and get that amount of DNA amplified so that we can actually see what's going on. So then we fix the DNA to a special type of filter paper in such a way that it's single-stranded and permanently stuck there. And so the single-stranded DNA is then available for hybridization to whatever we might want to try to hybridize it to, in this case, the allele-specific oligonucleotides that we just made. So we're going to design and label the two oligos, one for the wild type and one for the mutant. It doesn't matter which strand you're going to use, but it has to be exact matches to either one of those. And these are going to be labeled for the purpose of finding them later. Now, in the old days, they were radioactive, but now they would be fluorescent. So now we're going to hybridize these labeled oligonucleotides to samples from the people's DNA, and we're going to try to see what alleles that person has based on what fluorescent nucleotide stays stuck versus one that does not. Okay, so reiterating, the exact matches will hybridize and be visualized. Inexact matches will not form stable hybrids, and so the exact sequence that we see will allow us to genotype the person for that mutation. Okay, so this is not the kind of blots that are used anymore, but it is historically interesting. It was the first one ever, but they do illustrate the same idea, which is used currently, the idea of the allele specificity, that each oligo can detect its exact complement. So that is the basis of many, many ongoing PCR and sequencing-based tests. Okay, so here we are again. Here's our oligos. We've got a GAG versus a GTG, and these are 19 mers, meaning they're 19 nucleotides long, and they're single-stranded. Okay, this slide shows the result of an experiment in which we have DNA from 6 different people, labeled 1 through 6, and we're going to test the DNAs from those people, which have been PCR amplified and spotted onto the filter paper. And we're going to do 2 hybridizations, one to the wild type beta A allele, and that's the top row, and then we're going to hybridize them also to the beta S allele oligo, and that's the bottom row. And the pattern of spots is going to tell us what the genotype is of each of the people. So, for example, person number 1, their DNA hybridized very nicely to the wild type allele, giving a nice, very distinct dark spot at that position. But their DNA did not hybridize to the sickle cell allele. The spot from their DNA is blank. So we can interpret that to mean that that person is homozygous for the wild type allele and does not carry a sickle cell allele in their genome. Person number 2, on the other hand, does not hybridize to the wild type allele. They hybridize only to the beta S allele, so that both of their alleles are of the mutant type, meaning that they do have sickle cell disease. Person 3 is a heterozygote, right? They hybridize to the A and also the S, so they have one copy of the wild type allele and one type of the beta, one allele of the beta S locus too. Only 4 is fully wild type. Number 5 is a heterozygote, and number 6 has sickle cell disease. So by reading the pattern of spots, we can tell what the genotype is for the person, and very straightforward and simply. And this works because we can PCR, we know what gene we're looking for, and also we know the exact mutation that we're looking for, so that we can design the correct oligonucleotide to detect it. Okay, so I think I just said all this. After you hybridize and wash, you can read the genotype from the pattern of spots. Okay, which people are homozygous for the sickle cell allele? Number 2 and number 6. Person number 3 is heterozygous. Okay, so we can read the genotypes. This just spells it out. For some reason, they write this beta A, etc., as a way of representing this. Just You have to get used to the different nomenclature, I guess. Our next example is going to be cystic fibrosis, which is not at all a rare disease. About 1 in 30 Caucasians is a carrier, and there are approximately 1 in 3,500 births in the United States overall where the baby has some version of cystic fibrosis. Now, it's not restricted to Caucasians. People of all ancestries, ethnicities have this disease in them, sometimes different alleles, but this is a disease which is not strictly any particular ethnic group at all. It's also not particularly rare, and it's very important to identify people who have it so that treatments can be applied. Okay, the defect is it's a protein. The protein is a chloride ion channel, and it's needed to maintain water balance and chloride balance in some tissues. Important ones are in the lung specifically, as well as the pancreas and other sites. The lethal result of this disease is that thick mucus secretions build up in the lung, making breathing very difficult and leading the person prone to a lot of bacterial type infections that are difficult to clear from the body. Okay, the pancreas is also a very important target because the secretion of pancreatic enzymes is affected, and CF men are often infertile due to problems with secretions in the reproductive tract. Now, as classic CF has been considered lethal, but with newer treatments, people live much, much longer with much better quality of life. Okay, so the management includes antibiotics to prevent infection and physical therapy to keep the lungs as free of mucus as possible. Importantly, from the standpoint of this particular discussion, is that the severity of the disease does vary quite a lot according to whatever alleles the person has, as well as other genetic factors which are not really understood quite yet. Okay, so newborn screening, we know that newborns are screened for about 30 different genetic diseases shortly after they're born. This is not a DNA test in the case of CF. Oddly, the test looks for elevated levels of a protein called immunoreactive trypsinogen, IRT. So the basis of this is that CF babies have damage to their pancreatic ducts even before they're born, and that the damage to the pancreatic ducts leads to the accumulation of this particular protein. So looking for this protein is a very indirect method of detecting CF, but it's really quite effective and it's easy to perform on everybody. But once something happens, then the test is certainly repeated, and that's when you go on to include mutation panels and physiological tests such as sweat chloride. So as a first level, it's actually very effective, but it's not a strictly diagnostic of CF. Okay, in terms of the protein and understanding why there's so many alleles and what can go wrong, it is a cell surface protein of epithelial cells, a multiple pass membrane spanning protein, and it's a very large protein. And here's a picture of what it looks like. It's a chloride ion channel sitting on the surface of the epithelial cells, and what it's supposed to do is open and close in response to ions in order to maintain proper homeostasis of those ions in water in the cells. Okay, this is jumping ahead just a little bit, but if you envision what's happening, this protein is supposed to sit in the surface of the cell, but as a result of the defect in the ion channel, you have your decreased chloride secretion and the accumulation of this thick mucus. And shown outside the cell is the bacteria which can get trapped in the mucus and lead to recurring infections in these people. Okay, so mutations, the most common one is called delta F508, which is simply a deletion of a particular phenylalanine from the protein at that position. This is a very severe allele, and it's by far the most common in that about 70% of CF chromosomes have this particular mutation. But there are many, many other mutations which also can cause disease and can exist in conjunction with the delta F508. Okay, so DNA testing for CF is difficult because of all the possible mutations that can cause the disease. So many different alleles have to be tested at once. All right. So there was an early version of this which looked just for the delta F508, which of course, being the most common mutation, is a definitely good place to start. And so the early tests for this were just like the allele-specific oligos for the sickle cell, where you design the PCR primers to amplify the region of the person's DNA, spot the products on a membrane, and hybridize to the labeled oligos to detect homozygotes and heterozygotes. Okay, so here's a picture of that, and they really should have divided this particular filter into two, and they didn't. But it really represents just a test that looks exactly for that one particular mutation, which is a 3 base pair deletion, which removes one phenylalanine from the protein. Okay, so there's many other mutations. And 15 years or so ago, the American College of Human Genetics recommended a test where you actually test for 23 different mutations, any one of which or any two of which could be present in any particular person. And so this testing for 23 different things at one time sounds kind of difficult, but with newer technology, it's actually fairly easy. Okay, so this panel includes the 22 to 24 of the most common, not to say all of the mutations. And the technique earliest is what's called a reverse dot plot. It could be a microarray now, but a reverse dot plot is really a microarray in some sense. Okay, so the idea is if you're going to test a lot of things at once, you don't make a dot blot where you've got the person's DNA on the filter. You make a blot where the oligo is on the filter. So that's reversed because the probes, which are sitting there on the filter, are going to then attach to the person's DNA that you're floating in solution. And of course, the advantage to this is you can put many, many spots with many, many probes on it and test simultaneously for all of those in a very simple kind of way. Okay, in fact, the 23andMe microarray actually does include 23 of these mutations that are there that are recommended by these people. That's the SNP genotyping panel, which tests more than 600,000 different single nucleotide polymorphisms, but these 23 SNPs are actually among those. There are many more mutations that are not represented, however. Okay, so how to test for all of these at once, and it's kind of an overwhelming number of different things, and they come in different varieties. Usually your delta F508, of course, is the most common, but you have, for example, R553X. That's a chain terminator, obviously, because you've got your X, meaning that there's a stop codon there. So you do have some mutations which cause premature termination. You have single nucleotide or single amino acid changes throughout, but there are also changes in splice junctions and all sorts of things. So it's a standard collection of different types of mutations, but instead of looking for just one, you're going to look for, in this case, 23 of them. Okay, so the probes are the oligos on a filter or glass slide. The target is the DNA from the person to be tested, and that has been PCR amplified and labeled and hybridized to the array. Well, it's not that easy actually because the mutations might be present at many different places in the gene. So the person's DNA, when they're amplified, is not just amplified one region of the DNA. They may amplify as many as 23 different regions because you want to have the person's equivalent to those oligos that you can then hybridize to what's on the filter. But this multiplexing can be done not with 23 necessarily, but it can be done in batches and then pooled later. And these will be labeled fluorescently, at least in the earlier versions of this. So you hybridize your PCR products to the oligos and then wash an image and see which spots are visible. Okay, so this is something you have to think about a little bit. If somebody has, for example, they're homozygous for something, what we have shown here is the normal and expected mutant versions at some of these locations. So for example, the 1376N would be an oligo corresponding to the wild type allele at that position. And the one underneath it, G1376T, is one where we've had the single nucleotide substitution, which is the mutation that we're looking for. So we're going to have the wild type equivalents to all these locations, as well as the expected mutation which may occur in that person. So if somebody's homozygous, it's not so hard to see what's going to happen. For example, if you're homozygous for, say, the A95G mutation, we know that your DNA will match that A95G mutation, and you will have a dark spot where that is located. And you will not have a dark spot at the normal because both of your alleles have that mutation. You will have dark spots at all the other normal places because your mutation exists at that one location. The rest of your nucleotides are normal. So that's a little bit easier to see. What gets a little more difficult to see is when you have what's called a compound heterozygote, where the person has two different mutations, but they're both in the same CF gene, so they will have the disease in fact. So when that happens, they will hybridize to two of the mutant spots, one for each of their mutations, but they were also hybridized to the wild-type spots at those same locations. Now that's a little hard to get your mind around, but if you think about it, the protein, or think about the DNA sequence, for example, the A95G, if one of your chromosomes has that mutation, the other one doesn't. So the other chromosome would be normal for that, so you would hybridize to the normal spot. And say your other mutations, for example, the G871 A, your other chromosome would hybridize there, but its homolog would also have the wild type. So if you have a compound heterozygote, you would have two different mutations, but you would also have wild type equivalents at those same locations, accounting for the other chromosome. This is one way you have to kind of draw pictures for yourself to understand it, I think. Okay, so a baby that is a compound heterozygote for the A95G mutation and the G871A mutation will hybridize to C, mutant and normal prods for both of these sites. You have to draw a picture to convince yourself of that, I think. Yes, they are heterozygous for both of the mutations in the same gene. Okay, mom and dad of the compound heterozygote baby will most likely each hybridize to one of the mutant alleles and both wild-type alleles, each hybridize to both mutant alleles, each hybridize to both wild types but no mutants. One of the parents will hybridize to both of the mutant alleles and no wild-type alleles. Well, I think the answer is A. They will each hybridize to one of the mutant alleles, but they will also, on their other chromosome, have wild types. Yes, this one takes some thought. Okay, so things have advanced even more, and there are a lot of technologically complicated tests that basically do the same thing. There's a sequencing-based test which looks for 139 different variants, and there's also next-generation approaches, etc. These are not usually offered to people because usually the child will have one of the well-known mutations. So these are largely for research purposes, but it's nonetheless pretty impressive how easy it is to scan for hundreds, literally, of different mutations with the resulting from these technologies. Okay, so it helps to know about the genetics and to understand what's going on with the protein in terms of defining or really developing effective strategies. Okay, so the point of some of this is to develop therapeutic drugs that are directed at the effects of specific mutations and to try to counteract those specific mutations and what's gone wrong with their proteins. And some pretty good progress has been made in this fairly recently. Okay, so here's our protein again. It's a chloride ion channel, sits in the membrane, and it has to do a lot of things. And that's the basis of some of these drugs. Okay, the things it has to do, it needs to get synthesized without getting degraded. It's a membrane protein and it's glycosylated, so it has to make its way through the secretory system, through the ER, through the Golgi, in order to get to where it's supposed to go. So there's a lot of cell biology which has to go correctly to get this protein to its correct destination in the cell. It also has to function as an ion channel. So it needs to sit in the membrane correctly, bind to ions, and open and close correctly in response to them. So sometimes you can have mutations that the protein gets to the membrane, but there's something wrong with its ion channel function. And that, too, can be targeted by looking for small molecules in drug screens. So CF mutations have been classified, actually. This is a slightly older scheme, but the idea is that sometimes there's something totally wrong. If you've got a stop codon mutation, that will not produce any active protein. The processing block occurs in the F509 delta, where a lot of the protein gets degraded without ever making it to the cell surface. And that's obviously something you can target with a drug. Some of the alleles lead to blocks in regulation of the ion channel, conductance of it, etc. So depending on what's wrong, you can look for drugs that will fix that particular problem, ideally. Okay, this is just another slide which has a little bit more detail like that. Okay, so small molecules have been identified that will bind to these mutant forms and improve their function. Some of these have been approved by the FDA. Some of them can help the protein stabilize, which is to say the delta F508 mutation. There are drugs now that help the protein survive the pathway through the secretory system. It also, that mutation actually causes issues with the channel, but still you have to be able to get it there in the first place. So the new drugs, if you have the F508 mutation and the drug binds to it as it makes its way through the secretory pathway, if you can manage to get it to the cell surface, that will clearly be very helpful. Okay, there's other drugs that will bind to the ion channel itself, maintaining it in an open position longer and therefore facilitating its actual function. So the drugs are given in combination and have been really quite effective in helping people, at least ones with those particular mutations that affect those particular functions. And this can be targeted to whatever the mutations the person actually has. Okay, so correctors are ones that help the protein fold correctly, because some of these are a problem with folding. And others can improve the activity of the ion channel once it gets to the cell surface.